Hi, my name is Morgan Rockwell, and today I'll be talking about my research on the prevalence of Francisella tularensis in raptors and how hawk flies have a role in this. It is known that North American raptors can be infected with Francisella tularensis. For the rest of the talk, I will be referring to Francisella tularensis as tularemia, its more common name. Tularemia can be transmitted from the consumption of rabbits. Raptors have a high diet of rabbits and other small mammals, so they can contract tularemia from eating rabbits. It's also known that Hippoboscid flies are vectors that are found on North American raptors. So it is possible that the Hippoboscid flies can transfer tularemia to raptors that way. In my research, I had studied the transmission pathways of tularemia within the raptors and the Hippoboscid flies. I had tested this by performing a direct and indirect enzyme-linked immunoabsorbent assay, also known as ELISA. So what is tularemia? Tularemia is also known as rabbit fever, hunter's disease, deer fly fever, tick fever, and many other things. Tularemia is a gram-negative bacteria that can be found most prevalently on parasitic insects or vectors, also in ground mammals such as rabbits, rodents, etc. People who are in contact with these parasitic insects, such as people who go on hikes or lab workers and veterinarians that have contact from the patients that they work with, or with ground mammals, with the hunters, farmers, lab workers, or veterinarians are at the highest risk of contracting tularemia. Tularemia for raptors specifically can be transmitted via indigestion of rodents or other small mammals. It could be spread through bird-to-bird contact, or it could be spread through the raptors consuming contaminated water or contaminated food. In my study, I specifically use Hippoboscid flies or louse flies as my study of transmission. These flies are alga ectoparasites of birds and mammals. They're most likely found within birds. They are endotropic viviparity, which means that they give live birth one pupa at a time in the bird's nest. They also have a small flat body plan, so whenever they do grow to adults, they can shimmy their way up into the feathers of the bird and be able to infect the bird that way. For most of their life, they stay on the bird unless they are reproducing in the bird's nest or they are pupas growing from the bird's nest. This has a very small niche for them, and so they most likely are only in contact with the birds. Like I said, they can infect the birds from biting them and sucking their blood. That is how they live. So they spread many bloodborne pathogens. And we also know that tularemia lives within red blood cells, so it is likely that tularemia can be spread from blood-sucking insects. Here on the right, you could see the flat body plan of the Hippoboscid fly on a female and how you can see the abdomen has enlargement because of the pupa that's within it. Here, we can see more closely the transmission pathways of tularemia. So here on the right, you can see that the eastern cottontail rabbit can be infected with tularemia, and it can be passed on to people who either eat the rabbits, such as hunters, or it can be also, other studies have shown that if you are in a suburban area and are not hunters, that you can contract it if you have your lawnmower and run over baby rabbits and get it from that as well. Another transmission pathway is that if the raptor eats the prey and can contract it that way, and in my study today, we'll be studying to see if Hippoboscid flies can give great horned owls or any raptors in the study tularemia. Here's a short list of my materials and methods, which I'll go into detail. All of my blood samples for my lab and the flies were collected from the Illinois Raptor Center. My first test was a direct ELISA for flies. I had first, here you can see in the picture to the left, prepared the fly extracts by grinding them up and then coating a well plate with it. So with the fly antigen coated on the well plate, I then added my LPS antibody on top of that to see if there was any prevalence of antigens of tularemia. The second portion of my methods was a direct ELISA with the bird plasma. It's very similar to the same methods as the direct ELISA with the flies. Here, I instead coated the plate first with the bird plasma and then added the LPS antibody to that to see if there's any prevalence of tularemia inside of the bird plasma. And to see if the bird plasma, the tularemia antigens were present, we ran it through a plate reader, which you can see here to the left. The more yellow coloration there is, the more prevalence there is of tularemia. The last portion of my methods was an indirect ELISA. Here we first coated the well plates with LPS, and then we added the bird plasma on top of that to see if there is any bird plasma antibodies that would attach to the LPS. And then we added a secondary antibody to detect the antibody's presence. In the results, we found that the control flies that we had used had been validated for the LPS, so we could see that the direct ELISA was an accurate demonstration. And then for my direct ELISA, we had found that 22.9% of the flies had tested positive and 27% of the birds had tested positive. We also found that when we tested birds that were infected with positive flies, 59% of them had also tested positive for tularemia as well. We also found that there is no significant difference in the prevalence between flies and birds overall. Here on this chart, you can see which birds had tested positive for both flies and for the bird plasma itself. We can see here there was the great horned, or the barred owl, the great horned owl, and the red-tailed hawk. We can also see that if there was any positive flies on the bird, that there would always be positive tularemia within the birds as well. We also found in the indirect ELISA, we found that there was an overall prevalence of 14% within the birds with and without flies. We also found that the prevalence of birds with flies was higher than the overall prevalence. So that means that the birds with flies that had tested positive was more prevalent than just the birds itself that had tularemia. We also found that the birds that had tested positive in the direct ELISA, 73.7% of those had also tested positive in the indirect ELISA. Here's a chart for the indirect ELISA, and here you can see the highest prevalence of raptors that had tested positive for antibodies of tularemia were the great horned owl, the red-tailed hawk, and the turkey vulture. We also had records submitted to us from the Illinois Raptor Center that showed that birds that had tested positive for both the direct ELISA and the indirect ELISA had symptoms of lethargy, trouble standing, poor mouth color, and anemia. The birds that had tested positive for the indirect ELISA, 51.2% of those had no blunt force trauma, broken bones, and were not healthy orphans. And the same was for the direct ELISA, where it had 69.5% of those had the same symptoms. This means that they had some sort of illness whenever they were submitted. Records also showed that 46.5% of those birds that had tested positive in the indirect ELISA, and then 52% of those that were tested positive in the direct ELISA, were also considered to have poor mouth color. Records also showed that the birds that were submitted with skunk smell had tested positive for tularemia as well. So overall, we had found that there was a highest prevalence of tularemia found within the larger birds, such as the great horned owls, red-tailed hawks, and turkey vultures. This may occur because they have a very high diet in rodents, rabbits, and other small mammals, putting them at a higher risk of tularemia. And specifically, turkey vultures, they have a very high diet in carrion, so that makes it have a higher prevalence of infection for tularemia. We also found that there was a very high correlation of tularemia with the flies and the birds that have hosted with positive flies as well. This shows that the Hippoboscid flies play a very important role in the transmission of tularemia. Other studies I have found with Halos et al. Had a very similar findings. They found that Hippoboscid flies had played an important role in the transmission of Bartonella bacteria. This was very similar in the fact that there is a positive correlation with birds that had positive flies of Bartonella, with the birds also showing positive symptoms as well. They had a 84% prevalence of tularemia. Another study with the Levinsky, Bunon, and Sinclair found that Cooper's hawks, sharp-shinned hawks, and red-tailed hawks will most likely be parasitized, which we saw earlier that red-tailed hawks were one of the highest prevalence of tularemia. That these raptors were more likely to be parasitized with Hippoboscid flies. This may be because they spend a lot of time at their nest, and as a baby, they also stay close to their nest and not go out until later. We also found that most birds that had tested positive for tularemia in both the direct and indirect ELISA didn't have any blunt force trauma and broken bones, or were healthy orphans. Like I said before, this is because they were submitted into the Illinois Raptor Center with a record showing that they had an unknown illness that was not recorded. So these symptoms were likely to be tularemia. And this shows that the direct ELISA that we had used and the indirect ELISA were an accurate tool for testing to see the prevalence of tularemia within the raptor birds. Last thing we found was that birds that were sprayed by skunks were often positive with tularemia. It was 5 out of 8 of them. So this could be due to either these skunks having tularemia and transferring it from scratches and bites, not the actual spray itself. So through some kind of altercation, like the great horned owl we see to the left, or it was the idea that the spray itself may have impaired its ability to hunt by either making its eyes more sensitive and not being able to see, or the fact that the skunk spray was so strong that it was unable to get around the prey and actually hunt the prey as easily. So instead, it probably scavenged for either carrion or animals that were weaker and more likely to be infected with tularemia. So overall, the take-home message is that if you do see raptors that are submitted into a rehabilitation center that have been sprayed by skunk, just to make sure that you're cautious and wear your PPE, because it may have tularemia and it can be transmittable to either other raptors at your center or humans themselves if through scratches or bites. Another thing to keep wary about is if you have Hippoboscid flies on raptor centers that are submitted into a rehabilitation center, just to make sure that those Hippoboscid flies are all contained and are not spreading to other raptors that are coming into the center. And also if you are hunting or hiking or doing any recreational practices outside, or if you're in contact with small mammals or rabbits, to make sure that you are being cautious and wash your hands after, because it is more likely that you either get bites from flies that can cause tularemia, or if you're in contact with the small rabbits, that you also make sure that you are being very cautious of that. I would like to thank the Billings Field Study Fund and the Department of Biology at Millikan University for providing funding for my research project. And I would also like to thank the Illinois Raptor Center for the samples that were given to me, and then Dr. Wilcoxen for helping me perform the experiment as well. If you have any questions, please let me know.